Produced by Yue Liu.
Good morning, everyone. I am Yue Liu, a third-year PhD student in plant biology at Iowa State. I'm very happy to introduce our next speaker, Dr. Jinliang Yang. Dr. Yang earned his PhD in genetics from ISU in 2014. He is an associate professor in the Department of Agronomy and Horticulture at the University of Nebraska-Lincoln, conducting research in plant genetics and genomics. Dr. Yang's work focuses on integrating multi-omics data and functional annotations to bridge the gap between genotypes and gene phenotypes, thereby enhancing the efficiency of genome-wide association studies and genomic selection. His research has been supported by NSF, USDA, and DOE, and he has published over 40 peer-reviewed papers on journals such as Nature Communications, eLife, and PLOS Genetics. This morning, Dr. Yang will be presenting on the topic of leveraging multi-omics to review spatial and temporal adaptive genes to nitrogen limitations. Please join us in welcoming Dr. Yang.
Thank you, Yue, and thank you, the students organizing committee, for inviting me. It's so nice to be back. Yeah, it's about 10 years. I got education here at LSD University, working with Passion Novel. Now it's my honor to present the works we have done over the past several years using the knowledge I learned here. All right. So glad to follow Jamie's talk. She's working on legume, I'm working on corn. Corn is my favorite topic. She's working on phosphorus. Now I'm talking about nitrogen, another major topic. So nitrogen use efficiency become important, well, because unleaching, nitrogen leaching is an issue in the most of the area of the Midwest and also across the globe. So I'm using a multi-omics approach. Or actually, it's a population genetics and quantitative genetics approach, trying to understand through the evolution perspective. So what's the nitrogen? Well, how plant adapted to different nitrogen conditions from time and across space. Nitrogen is essential micronutrients for most of the living organisms, especially for plants. Plants actually all under very tight nitrogen budget. So to cope with this tight nitrogen budget, different crop species and different plant species adapted using different strategies. C3 crops like legume, soybean, for example. So their strategy is to work with microorganism. So they form a partnership with the microorganism. So let the microorganism to fix nitrogen for them. So therefore, this cycle is sustainable. But we know C3 crops, cold season crops, so their nitrogen, their photosynthesis nitrogen use efficiency is relatively low. C4 crops, such as sorghum and maize, so their photosynthesis nitrogen use efficiency is very high, meaning that they can converting the solar power into starch, which eventually treated as the yield, and the efficiency is very high if we provide them with sufficient food. For plants, that's nitrogen. So the industrial fixation of nitrogen make it possible to produce large quantity of inorganic nitrogen fertilizers. So this will be injected into the soil, provide food for these C4 crops to grow and produce carbons as calorie sources for animals and human. But that process being chaired for quite a long time. We even awarded Nobel Prize for this invention, for its breakthrough. But later on, we realized that by overusing the fossil fuels, we actually create serious environmental problems, ecological problems, including leaching, soil contamination, water system contamination, and also lead to the greenhouse gas emission. So even worse, so this agriculture ammonia usage actually emits a lot of ammonia emission, actually contribute a lot to the fine particle air pollution called PM 2.5. So this data shows that the severity of the N shares of the PM 2.5 air pollution across the globe. You can see from this graph that so across the world, especially in agriculture production areas, Midwest, Europe, part of China, the air pollution due to ammonia emission is very severe. And the cost of this agricultural production for PM 2.5 pollution costs in total 23 million years of life loss in 2013 alone. So that's about 1 day per person per year on average. So the associated healthcare loss is about 400 billion. So that's about $6 per person per year. So therefore, so this, I didn't make this up. This is reported by this Gu et al. Paper published on Science. So they raise this question and they provide their solution. So their solution is that from an economic point of view, so they suggest to cut the emission due to ammonia application in agricultural production by half, 50%. So they argue, well, if we do that right now, so the benefits actually is over 150 billion, but the cost is only about 50 billion. From so agricultural perspective, that's not possible because we need to feed the world, right? So if we cut the ammonia application, the agricultural production gonna be lower. So that's we shouldn't calculate it that way, but our calculation, previous calculation for the farming profitability is like this way. So we consider the nitrogen output, which is the yield. We consider the nitrogen input. We subtract the nitrogen output by the nitrogen input, but without considering all those environmental costs. Based on the previous calculation, this should be calculated into the profitability. So that actually led us to revisit this yield improvement trend. So this probably we have showed this a number of times, the yields being increased for corn production over the past 100 years, while breeding started from the 1920s. Genetics contribute a lot to this, and also agricultural practice and statistics, all environmental designs all contribute to this yield improvement trend. But if we factor in the environmental costs and also subtract by the nitrogen application cost, so this is using this simple calculation. So if I assume it's about $4 per bushel and the $1 per usable pound of nitrogen and charge with $2 tax on the per pound of usable nitrogen. So actually the yield, the profitability trend become ugly. we barely, nowadays, we barely better off than the poor agriculture industry era. So in terms of the societal benefits in general, if we consider the environmental cost. Well, from a global point of view, across the world, so I'm including all those urban areas, glacial regions, and heavily industrialized crop soils, for example, the Midwest. So there's still 20% of the natural territorial regions that experience nitrogen limitation and also phosphorus limitations as well. So the nitrogen limitation is my focus here. So you can see from this global view, so nitrogen limitation show a latitudinal and also elevational pattern. So meaning that north, high altitudes usually be more likely be nitrogen limited, and the higher elevation, highlands especially tend to be unlimited. That's for a reason because nitrogen in the natural environment through mineralization using microorganisms. So higher elevation, less vegetation, therefore less microbial activities, therefore less nitrogen mineralization. So I'm talking about it's the highland areas, the high elevation areas. So there's a severe nitrogen limitation, but nevertheless, maize historically adapted to those areas. So we know that maize domesticated from lowland Mexico around 9,000 years ago. A recent study by Rasibara et al. Found that about 6,000 years ago, that lowland maize subspecies of glumis were integrated with another maize subspecies, Mexicana, which is a highland tucente. And they mix up together, so some highland genetic materials integrate to the lowland materials and then adapted to the highland areas in Mexico. So before it spread to much of the Americans. The highlight here is the Mexico highlands. That's the maize historically adapted to this Mexican highlands. And then after this adaptation, so they spread out to the Andes highlands. About 2,000 years ago, they reached to the North America, reached to the Colorado Plateau highlands, stayed there and adapted about 1,000 years, and then spread to much north. So that's about the domestication history. And then post-Columbia times, it spread out to Europe and then South Asian, African, all across the world. So adapted to all those geographic regions with some of the regions with sufficient nitrogen in the natural soil, others with limited nitrogen in the natural soil. So as a species, so in a natural environment, it evolved in such a way that they adapted to the variable nitrogen limitations. So that's a system I try to leverage. So, I mean, while I'm studying a crop, but this crop can also serve as a model species to study the plant adaptation to various nitrogen limitation conditions across time and space. So first of all, let's take a look at the timeline. So this breeding, so actually the left panel shows the breeding history. Breeding started from 1920s. As I highlighted earlier, nitrogen fixation is only started around 1960s. That's the time for the Green Revolution. So maize actually was started breeding way before 1960s. It started around 1920s. So previously, the breeding was conducted under nitrogen limited conditions. And later on, after 1960s, especially after the 1970s, it started to be under nitrogen sufficient conditions. We oversupply the soil with sufficient inorganic nitrogen fertilizer. So therefore, our first hypothesis is that the recent maize improvement under nitrogen oversupplied conditions have resulted in the loss of function of the critical nitrogen resilient genes. To test this hypothesis, I get some old error lines, meaning the lines developed, the inborn lines developed before 1960s, and the new error lines developed after 1960s. What we know, line actually take years to develop and release. So to be safe, we selected only lines developed after 1970s. So know that BSAM3 was developed after 1970s. That's released at around 1972 here. So at our state. So first of all, we planted those new error and old error inborn lines under a same environment, but with different nitrogen treatment under loan conditions and sun conditions. So we compared the phenotypic performance here and measures the leaf nitrogen level at the adult stage. And you can see, so in this figure, on the left-hand side, there's a nitrogen level differences for the new error inborn lines and the old error inborn lines under high-N and low-N conditions. So when I calculate the ratio, so high-N over low-N ratio, so we found that there's significant difference. So you can see on the right-hand side, the new error inborn lines performs significantly better under high-N condition, which makes sense because what we selected in the past 60 years also is nitrogen responsive lines, meaning that we want high yield under sufficient nitrogen. While on the other hand, the old error lines are actually more resilient, meaning that you add more nitrogen, it doesn't respond much. Or on the other way, think another way, meaning if we reduce the nitrogen application rate for the old error lines, it didn't change much. It's more resilient. They don't respond to high-N, but they're more tolerant to low-N. So that's a resiliency trait we are trying to find. And this phenotype actually consistently observed for many other traits we observed. Leaf chlorophyll and leaf dry weight and the leaf fresh weight and others I didn't show here. So we then go ahead to conduct the genomic selections again, using the genome sequence data from the old error inborn lines and the new error inborn lines. So we found this striking peak on chromosome 5. And so break down this region, so we found there were 6 LD blocks, namely LD block 1 through block 6. So the most pronounced signal located on block 4, right in the middle. So we about hypothesis this will likely be due to population structure, and then conducted a population structure analysis only using this block 4, LD block 4, and found, well, there's 3 haplotypes within the LD block 4 in general. It's not perfect, but there's 3 clusters in general. The new error cluster, new error haplotypes, and old error haplotypes are also recombinant haplotypes. And each of the haplotypes composed of the materials from different subpopulations of maize, suggesting, well, this pattern is not driven by population structure. Well, encouraged by this, we then start to further phenotype those in the well-controlled environment with a much larger population. Here we use a maize association panel composed of 300 diverse maize lines in the Innovation Campus greenhouse at UNL. So this LAMTech technology enables us to characterize the phenotypic traits across the whole developmental stage of the maize. until maturity. So we measured, well here, so I've only show you the reliable trace. I'm not showing all this image extracted trace. So we manually measured 18 leaf physiological trace while planting. So this is very expensive, $3 per pot per day. So therefore, we try to ensure every pot have a plant in it. We plant 3. So we thin the plant before so after they germinate. So for this seedlings that grow about 2 weeks, we actually also sample the seedlings to measure some other traits, metabolized traits. We focus on this region and we found, so using this population is also associated, that region, especially the LD block 4, also associated with some other traits. And well, again, we confirm it's associated with the leaf nitrogen content, but also leaf angle and leaf area and leaf dry weight. So as I showed earlier, so we also measure the metabolites. So interestingly, we found that this LD block 4 also associated with the metabolites, especially you can see the new error haplotype show a higher levels of the secondary metabolites, for example, lysine, that has an N element in the formula. But for the carbon type of metabolites like fructose, not only this one, this is just an example, but a number of those following this pattern, that the new error lines for this carbon-like secondary metabolites, the new error lines actually have lower level, but the old error lines have a higher level of this carbon-rich metabolites. But then a larger GWAS. So this using this GWAS, well, so this is the published data. We just did a reanalysis on using this published data of a nested association mapping population, which composed of more than 4,000 invariant lines. We be able to identify again a signal in this region located on chromosome 5, associated with the leaf chlorophyll content. So if we zoom into that region, that arrow actually, that's the SNP associated with the chlorophyll content directly hit a gene. And after we take a closer look at the gene, so we found that there's a cluster of 3 of them that show a sequence of similarity. Those 3 genes phylogenetically close to Rhabdopsis glutamate-like receptor, GLR. So we name them arbitrarily as the GLR 3.4, A, B, and C, based on their order. So as you can see here, so the 2 genes actually is located within the block 3, LD block 3 and LD block 4. So especially for the LD block 4, which is the region, we have GWAS evidence, we have a selection evidence, and we have association evidence for a number of different traits. And also check the gene expression level for these 3 genes. And found the second gene, which is the GWAS signal direct hit gene, that show clear differential gene expression between new area invariant lines and old area invariant lines, and is highly expressed in adult leaf tissue. The third gene also is highly expressed, but there's no difference between new area and old area invariant lines. The first gene is lowly expressed, likely be a malfunction gene. And using the gene expression obtained from, again, published data from Buckler et al. So we found this second gene, GLR 3.4, likely be a cis-regulated. There's a strong EQDR signal just located within the genic region. We also check the Chinese lines to see what the allele frequency changes over time. You know that in China, modern breeding started after the cultural evolution. So that's after 1960s, after the Green Revolution. So you can see the yellow line indicates the allele frequency changes for this new era imbalance, a new era haplotype. And so in Chinese lines, those are 400 Chinese elite lines released from 1960s all the way to 1910s. So their allele frequency changed steadily from about 10% to about 70%. Not being fixed yet, but steadily increase. So further, we checked the allele frequency, a new era allele frequency in two sentient population, the maize wild ancestor and the masculine races, and the old era and new era. You can see, so this even in two sentient, so that new era allele is present. Is so suggesting this is not a new mutation, it's from a standing variation. An increase in the land races, decrease a little bit in the new era, and rapidly increase in the new era. Likely response to varied levels of the nitrogen limitations. Well, nowadays we have this de novo genome assembly. So in maize, there's about, I lost my track, probably about 100 de novo assembled genomes. This the analysis done about 2 or 3 years ago. So at the time, so we checked the some imbalance with the de novo genome sequencing data that released in the new era and the old era. So we checked the structural variation of these three candidate genes, A, B, and C. Interestingly, we found that second gene, the strong candidates, show a large structural variation. So there's a transposon insertion and deletion in the new era and the old era, in the intronic region. But the first one, the third one, there's not much large structural variation. So again, closer look at the second gene, our strong candidate. So the insertion likely be degenerated transposon. So there's not intact transposon. So partially, there's some likely be nested insertion. Well, this suggests again, so this insertion is a historical event, meaning that both alleles have been kept in the population for a longer time, maybe can trace back before domestication even happened. So we also checked this again, the second gene. So the transposon insertion, especially at the third intron. So that's highlighted by the red triangle there. That's a transposon insertion into B73. So we found that there's a clear methylation differentiation between new era and the old era imbalance. So for the new era, so with the transposon insertion, their gene expression is high, and also their methylation is high. So this is counterintuitive. So methylation usually been considered silent gene expression, but this gene, the body methylation, actually associated with high gene expression. And for the old era imbalance, you can see the methylation level close to 0. So methylation is like that. You either methylated or not methylated, either 0 or 1. So this clearly shows that pattern. Well, you wonder, well, let's do some genetics. Well, in maize, genetics is not easy. So this analysis, computational analysis done, like I said, 3-4 years ago. And we are trying to using traditional reverse genetics to confirm the gene function, but that's not easy. So this is the whole timeline show the work we have done by crossing, identify the uniform mutant because we have to clean up the background, so we have to backcross and self. Last year, so we got this BC2F3, and we planted them in the field and treated them with high nitrogen and low nitrogen field conditions, and observed that the mutant little actually show a higher plant height compared to the wild type. So this is the data. So each with 2 replications and under high and low condition. Very interesting. And for the kernel, and you can see, so under low conditions, it didn't change much. So meaning that, again, consistent with our resilient hypothesis. So under low condition, the yield, well, the yield component, this is not yield, this is just a kernel size. The kernel size under low condition didn't change much, but under high condition, the mutant actually performs better than the, really better than the wild type. So again, the old arrow alleles seems resilient, but new arrow alleles seems more responsive to high nitrogen treatment. So here's the data. So know that this is the third gene. So this is not the strong candidates. This is 3.4C. So the genes highly expressed response to nitrogen, but there's no new error and old error differences. That's not the gene 3.4B that we are really interested in. And the conventional genetics is too challenging. But nevertheless, we backcross it again to further purify the background, just get the BC3F3 this year, 2 months ago. So we planted them in the greenhouse. And the seedling traits show the consistent phenotype that the mutant line actually be more resilient. As you can see, so the mutant line here show a higher phenotype, better phenotype. So especially under low conditions. So under low conditions, the seeding has color, and the seeding fresh weight is higher, and also seeding dry weight is higher. And the ratio again, so is significantly different for all those 3 traits. So we also want to get some the second gene knockout. So now we switch to CRISPR. Because these 3 genes are highly homologous, so we'll be able to design guide RNA to target all those 3 genes at the same time. We get a single mutant, double mutant, and a triple mutant. Well, this enables us to design a larger experiment in the greenhouse with 8 replications under high and low conditions, and we observe the seedling phenotypic performance. So on the left-hand side, that's a wild type under high and low conditions. And so here I highlighted the single mutant and double mutant. Some of them show significant difference, especially under low condition. For the triple mutants, a lot of those knockout mutants show better performance under low conditions as compared to the wild type. But we didn't see much differences under high conditions. So look at the ratio again, so resiliency. So a lot of those triple mutants show significant difference. So further, so we start to look at the mechanism. What's the possible reason of the genes that leading to this nitrogen resilient phenotype? We did this protein-protein interaction network analysis. As you can see here, so this glutamate-like receptor, GLR genes, are put right in middle of these 2 clusters. One cluster, want to highlight, is the nitrogen pathway. So a lot of nitrogen genes, glutamate synthesis, nitrite reductase, cluster there. And another cluster is ion signaling pathway genes. Sodium transporter, potassium transporter, calcium ion bonding gene. So suggesting, well, this gene might be connecting the nitrogen pathway and the ion signaling. While observing the phenotype, so we found this, especially under stress condition. We put them in a sandy soil, and we found this leaf tip burning phenotype. And so put on the regular soil with sufficient nitrogen, it goes away. We didn't bother much. But later in the season, well, for those days, if you guys still remember, this winter, some days that's extremely cold, -20°C. So, well, my plants, especially the triple mutants, get very stressful. They show this phenotype again. So I talked with some of my colleagues. They suggest this likely be calcium deficiency phenotype, and they put a calcium fertilizer. While this phenotype got recovered, we'll be able to harvest seeds from those mutants, stress the plants. Well, as you can see, so on the right-hand side, the white type didn't under any stress at all, but my mutant almost died. But we got it recovered by applying the calcium fertilizer. So that's actually consistent with the protein-protein international interaction networks that this gene might play a role in signaling. Well, so this research tells us that this glutamate-like receptor gene is not only present in plants, it's highly conserved across different species, even in mammal. So in human, actually, this nerve, so central nerve genes that this gene was believed to can combine with glutamate. So there's a cross-memory parts. These parts, if you combine with glutamate, this changes the conformation of the cross-memory part, open up the iron gate. So this cross-memory is the iron-gated channel, iron channel that let the signal pass through. In plants, especially in Rhabdopsis, there are some studies suggest that this GLR is a broad spectrum amino acid sensor. It's not only glutamate specific. It can interact with the broad spectrum of amino acids and open up the gate, let the signal to pass through, activate a series of downstream, possibly nitrogen pathway gene. So this experiment suggests that the subcellular localization of the GM GLR 3.4b and C actually localized in the memory. Well, so by checking this historical breeding pattern, so we identify these genes likely play a role in sensing the nitrogen level at a cellular level. And hopefully this gene can be utilized in breeding to increase or at least not decrease yield a lot under nitrogen conditions. Well, that's the spatial pattern I leveraged, the temporal pattern I leveraged to study this crop adaptation to unlimitation conditions. Well, there's also spatial adaptation. So I talked about during my introduction. So this allows us to further hypothesize that the naturally adaptive alleles in different latitudinal and elevational regions with different nitrogen limitations could also be possibly harnessed to bring the low N input agriculture and enhance the nitrogen use efficiency. So with that idea, and especially the glutamate-like receptor genes have been historically present in the maize genetic pool, trace back to even before 270. So this allow me to hypothesize that this pattern of historically maintained alleles likely be under balancing selection. So balancing selection maintain different alleles at the selection site. So according to Han et al., so this balancing selection gonna leave a genomic signature in the genome because the genealogy of the balanced selected loci gonna be stick around in the genome longer than you expected, than the neutral size. So if you two alleles being maintained in the population, so that'll have more chance to accumulate mutations to make that regions more diverse. So if you just scan the genome. So for example, this ADH locus, so it's under balancing selection. So this results in very higher diversity as you expected. Theoretically easy, but this signal is very minimal and is very sensitive to the materials you use for scanning for it. And there's not a lot of studies being very successful in identifying balancing selection signal. Recently, Chen et al. Developed this strategy called B2 statistics. They use this B2 statistics to model the sequence spectrum of alleles under neutral and balancing selection. They consider this as a mixed distribution. So if there's a balancing selection signal, so they're gonna show this little bump in the middle, and also the mix with the neutral size to form this mixture distribution like this. And we use this approach and scan the 270 population because we believe, well, maybe, so transposon insertion gonna make this balancing selection signaling detection is very complicated, make it even harder. So therefore, we believe our 270 population might be leveraged to detect the selection signal for the chromosome 5 GLR region. Well, we're not very successful. We detect a weak balancing selection signal on the GLR chromosome 5 locus. That's not very encouraging, but we know the reason because there's transposon insertion that, so the selection scans are leveraging the SNP information, but structural variation make it very complicated. So therefore, I talked with Adam and asked him to give me some elite materials that adapted to the natural soil. It's not in the industry, agricultural industrialized areas. These are the elite materials developed in the Andes, in the highlands, and in the tropical areas in Africa with low and unlimited soils. And this is also gem founder lands. So some of these lands probably only have 10 seeds left. Thank you, Adam, provide me those precious genetic materials. I sequence them and also backcross them. Well, so there's a bee gem populations by crossing this tropical founder lands to the beach B47 and the BHD51 to have this bee gem stiff stock and non-stiff stock. So we backcross this bee gem stiff stock, bee gem non-stiff stock, add up together, those are 300 different lines. For the founder lines, those are about 50 lines. Those are the lines very unique as compared to all the different global maize panel. So we published earlier. So this is the population structure of this data. So first of all, I'm just using the founder land, 50 founder lands. So there's we identified very interestingly a number of nitrogen related genes. if you take a closer look at this, we identify that GLR again, but this is a different GLR. So this is GLR. So our previous GLRs here, 3.4. Here we have a GLR 2.7 A and B, again, clustered together. Very interesting. And we also identify glutamate ammonia ligase gene, nitrate reductase protein, and some calcium signaling gene. So you may also wonder what those signals are. We actually have a notation for all those signals. Some of them make perfect sense for balancing selection. So for example, this one's associated with a small kernel and being cloned previously. Or to further validate those, so balancing selection just tell us that those are regions under selection, so they didn't tell us what trait they likely be contributed to. So to understand that, so we leverage the BGEM population we just created by backgrounding to 2 recurrent parents and put them in the field under lone conditions. Each with 2 replications. I'll go through this quick. We collected above ground traits and below ground traits, including the root associated microbiomes. I don't have time to talk about that. That's a whole different topic. So we measured the yield component trees and below ground root trees. So we found those trees, as expected, show heterosis, and the heterosis levels are different and high and low conditions. And well, I want to highlight one point is that under low conditions, the heterosis of root trees actually is more significant than the heterosis under high conditions. And the microbiome diversity is measured by Shannon diversity. So this is a measurement of the microbiome diversity. So inbred, as you can see, under low conditions, more diverse as compared to the inbreds and higher condition. And the hybrids actually have a very lower microbiome diversity as compared to inbreds, because, as you know, hybrid doesn't care because they don't need a microbiome to help, especially if the nitrogen is sufficient. So this is also consistent with the previous observation. All right, so and the microbiome also show heterosis. And finally, so I'm not using this for other purpose, just use this to cross-validate the balancing selection signals I identified previously. So this balancing selection signals that overlapped with the GWAS signals using the trees we collected over the past 3 years under high and low conditions. So some of this may be able to connect with the trait. For example, this signal is well, based on notation, is a kernel size related gene, but we found it's also associated with one microbe under low condition. And this one is associated with 20 kernel weight yield component trait under low condition, and this locus contribute to this trait's heterosis. So especially the combination with the tropical allele and the temperate allele as compared to temperate allele alone. So suggesting these tropical alleles have value to be rescued to enhance the nitrogen use efficiency. Well, to summarize, so I hope this talk will deliver a clear message that so in the future, tomorrow's agriculture, we really need to rethink about the direction of plant breeding. And my goal is lower the nitrogen input. So I won't read this. Basically, so I have further hypothesis. Well, this is the pathway, nitrogen pathway. So I have further hypothesis this GLR play a critical role involving in calcium-dependent nitrogen signaling. Is that I would like to thank my awesome team. So this without them, none of this can be done. So especially Dr. Genxi, an awesome postdoc who did all the computational work. also many other students' contribution, and my collaborators and the funding agency. Thank you very much. I'm happy to take any questions.
Hello, my name is Shelley Kinney, and I am this year's poster chair for the Symposium. I would like to thank the speakers for the amazing talks so far, so thank you. I would like to announce that the first poster session of the day will be starting at 10 a.m. with all even-numbered posters presenting. So please be sure to wander around and see all of the great posters. Thank you.